Claudia Moreno. Claudia Moreno, who's an assistant professor in the Department of Physiology and Biophysics here at the University of Washington. And she's going to talk to us about mechanism behind the dysfunction of the cardiac pacemaker.
Yeah, good morning. Thank you so much. Just let me start my presentation. So are you seeing my slide? Yes.
Yeah, we're seeing it just fine.
Perfect. So good morning, everyone. Thank you for being here, and thank you so much for the organizers by the invitation for participating in the Geroscience Symposium. For me, it's a real pleasure to be here today, sharing with this amazing Gero enthusiast community some of the things we are doing right now. In our lab, we are interested in understanding the mechanisms behind the dysfunction of the cardiac pacemaker during aging. For those of you who are not familiar with the cardiac pacemaker, which is an amazing tissue, I'm going to start by telling you a little bit about it. The cardiac, the human heart beats at about 100,000 times a day, and near 2.5 billion times in an average lifetime. And each heartbeat is initiated right here in the upper part of the right atrium in a region known as the cardiac pacemaker. The cardiac pacemaker is formed by specialized cells that have the ability to regenerate the spontaneously action potentials, generating the initial electrical signal that propagates through the heart to trigger heart contraction. The pacemaker generates this signal over and over again. Since the moment the heart is formed in the early embryonic development until the very moment we die. How fast our heart can be is determined by two factors. The first is how fast the pacemaker cells can fire, and this is called the intrinsic pacemaker rate. And the second factor is the autonomic control of the heart. In fact, the sinoatrial node, or the heart pacemaker, is the most highly innervated region of the heart. And it receives terminals both from sympathetic neurons, shown here in green, and also from parasympathetic neurons, shown here in blue. Under physiological demands, like the flight or fight response triggered by stressful or dangerous situations, the sympathetic neurons release noradrenaline right here in the pacemaker to accelerate the firing rate of these cells and therefore the heart rate. In an opposite way, the release of acetylcholine from parasympathetic terminals over the pacemaker deaccelerates the heart. Despite the reliable machine that the pacemaker is, unfortunately, this exceptional tissue is not exempt of the deleterious effects of aging. And as you can see here, actually, both the resting heart rate and the maximum heart rate of our heart changes with age. In the case of the maximum heart rate, there is a continuous decline from birth to old ages in our maximum heart rate. And this is mirrored by a linear decrease in the intrinsic pacemaker rate. What that simply means is that as we age, our pacemaker cells become slower. And this happens in every human as we age. In some cases, it can also lead to the onset of a group of pathologies known as sick sinus syndrome, which is a group of idiopathic diseases which main risk factor is age and is responsible for most of the artificial pacemakers that are implanted globally. Worldwide, there are more than 600,000 artificial pacemakers implanted every year. So the fact that the intrinsic pacemaker rate declines with age can be better appreciated when we isolate cells from the heart and study them without the influence of the autonomic nervous system. So here you can see some representative traces of action potentials that are being spontaneously fired by pacemaker cells that were isolated from mouse hearts. So we use the patch clamp technique to go and record the electrical activity of these cells. And as you can see here, we're showing in white the spontaneous activity from a cell from a young mouse, and then in red, one from an old mouse. And you can see how there is a slowdown in the frequency that can be summarized here in this plot, a significant reduction in the spontaneous firing rate, and also some failures of these cells to fire between action potentials. So why the pacemaker cells become slower with aging? This is something that is still poorly understood. And today I'm going to show you some of the evidence we have gathered about how aging is affecting important components in the function of the pacemaker. And I will show you how aging affects the activity of calcium channels, some ion channels that are really crucial for the function of the pacemaker. Also how aging affects the organization of these channels, and I will tell you why it is important to look into how ion channels organize. How aging affects the adrenergic modulation. This adrenergic modulation is what makes our heart go faster, and so I will show you a little of the evidence we have on what aging is doing. And at the end, if I have enough time, I will briefly show you some new results that we have about how aging is affecting the architecture of the pacemaker tissue. So if we go to look closer, what is happening here in the action potential between young and old mice, something that is observed, and I am imposing the two traces here. And in this zoom in, you can see that this exponential phase, that is the initial rise of the action potential, becomes slower in old animals compared to the young. In this diagram, you can see that this purple exponential phase corresponds to what is called the late diastolic depolarization. Both the early and the late diastolic depolarization are phases of the action potential that are crucial for the automaticity of the cardiac pacemaker. And as you can see here in these plots, both are highly affected by aging, so age causes a reduction in the velocity of these two components. To understand what are the ion channels that are implicated in these phases, I'm gonna just use this diagram where I color-coded some of the most important ion channels that participate in the generation of the action potential of the pacemaker. And I'm gonna ask your attention here. In the CAV 1.2 and the CAV 1.3 channels, these are members of the L-type calcium channels, voltage activated calcium channels, and these are the channels that I'm gonna mainly focus in this talk. Both of them are implicated in the late diastolic depolarization, but also the obstruct, because opposite to ventricular cardiomyocytes or neurons, the action potential of pacemaker cells is carried completely by calcium. So I want you to think that calcium is like the important ion that we care about here. So the question is, does aging affect the activity of calcium channels? And here, what we use is, again, electrophysiology recordings. We isolate pacemaker cells from young and old animals, and this time we use the voltage clamp technique to measure not the spontaneous action potentials, but this time we're measuring how much calcium gets into the cell by these specific ion channels. So the downward deflection that you are seeing here corresponds to how much calcium gets into the cell, and you can see that in old animals, there is a big reduction in the amount of calcium that we get. And this is summarized here in this plot. So is it possible that aging is affecting the expression of these channels? And therefore, that's why we have a reduction in the current. So we evaluated that using Western blots, but as you can see, interestingly, we saw the opposite. In old animals, it seems to be an increase in the expression of both the CAV 1.2 and the CAV 1.3 channel. And so what is happening in old animals that we have this reduction in the current? And here I'm going to talk briefly about something that we have studied in the last few years, and that is not related with aging, but more with the function of these calcium channels. So we have discovered that calcium channels have the ability to couple to each other physically and modify their activities. So under hyperpolarized voltages, calcium channels open in an stochastic manner, so independently, and they let calcium in. But if there is enough calcium in the vicinity of the channels, there is the activation of a protein called calcium calmodulin that bridges an interaction, a physical interaction between the C-terminal, creating the cooperative gating of the channel. So now the channels open at the same time, they open for longer times, and that results in an increase in the calcium that is getting into the cell. We have demonstrated that this modifies the activity and the way the ventricular cardiomyocytes fire the action potentials, and also it's a mechanism that is regulating firing frequency even in neurons. So something that we know is that for this coupling to happen, channels need to be close. And we have used super resolution microscopy to study in detail how ion channels organize and why this is so important. So this is a representative fluorescent image of a pacemaker cell that was stained against the CAV 1.2 channels. And this is the kind of image that you get under conventional microscopy. So you see all these light diffracted spots, and it's very difficult to see how channels organize behind these blurry spots. But if you use super resolution microscopy, then you can get this kind of resolution. You start resolving individual channels and can in detail understand how channels organize. And in fact, we have done this for multiple ion channels, and now we know that for ion channels, being clustered is the rule. So you can see here calcium channels packed into dense clusters of different sizes. And we wanted to know if aging is affecting the clustering of the calcium channels. So we, in fact, saw that that's the case. We saw when we compare the clustering of CAV 1.2 channels, and this is the average area for the clusters, you can see that in all animals, there is a reduction in the density of these big clusters and a reduction in the size of the clusters. Ion channel clustering is not only important for the way the channels interact with each other, but it's also important for the interaction with other molecules that regulate calcium channel activity. And we are particularly interested in understanding what's happening with the interaction with beta-adrenergic receptors. So as you remember at the beginning, I mentioned that beta-adrenergic receptors are the mediators of the adrenergic response. So this is what makes our heart go faster. Noradrenaline binds to either beta-1 or beta-2 receptors in pacemaker cells and can facilitate calcium channels to increase calcium entry, which results in an increase in heart rate. So to study how aging was affecting this signaling, we use a specific agonist to activate the beta-2 or the beta-1 adrenergic receptor. And in this plot, I summarize, I summarize the data. Oh, okay. Okay. So in this plot, I summarize the data. And what we are seeing here is, again, we are using patch clamp to record the calcium currents. We are looking how much calcium get into the cell. And then we compared this, what we call calcium facilitation, is how much of that current is increasing relative to this baseline. So here we apply an agonist that activates the beta-2 adrenergic receptor, and we see how calcium channels start increasing their activity in young animals, but this increase is significantly reduced in the old animals. Then we apply an agonist that is activating the beta-1 adrenergic receptor, and calcium channels get facilitated. And in all animals seems to be a compensation. Now, in these cells, the beta-1 adrenergic receptor is able to reach the same levels of facilitation that we get in the young. So we wanted to test the hypothesis that aging was altering both of these signaling, and we use proximity ligation assay. That is basically you perform a PCR reaction between the two proteins of interest if the proteins happen to be in close proximity. And you can see here how in the case of beta-2 adrenergic receptors, there is a big reduction in the proximity between L-type calcium channels and beta-2, and we didn't observe that for the beta-1. And actually, when we look at the summarized data, you can see how in all animals, this correlates very well with our current data. There is an increase in the interaction with the beta-1 receptor, which and a reduction in the beta-2 that results in poor modulation. So we have so far, and I showed you briefly that aging causes reduction in calcium current, alterations in the clustering and modulation by adrenergic receptors. And we also are studying now beyond the cell how aging affects the pacemaker. This is a fluorescent image of a clear tissue of the pacemaker, and this is how the pacemaker looks. And this architecture is important for the generation and propagation of the calcium signals. And I am going to show just these final two slides to give you a sneak peek of what we are doing right now and seeing how aging dramatically affects the architecture of the pacemaker and also the vascularization of the pacemaker, having a big impact in the perfusion of the tissue. So I will finish just by acknowledging the people from my lab, Sabrina Choi, Matias Baudot, Liz de la Cruz, and my close collaborator, Oscar Vivas. And under the current circumstances, I also feel obliged to acknowledge two people that are two young ladies that are not related with this project or science, but that have been taking care of my kiddos so I can survive this hard work of being a parent and working remotely and taking care of kids at home. And I think that's a problem that many of us are facing right now. So I wanted to just thank them as well. Thank you.
All right, thank you so much. It's fascinating work. We have one question from Nikolai Bonayeski. Do you know turnover rate of calcium channel in pacemaker cells? Do they ever get turned over?
That's an excellent question. And that's something that it is not known. And I think is really relevant to know. We want really to find ways to measure turnover with the idea that there might be problems in the trafficking insertion and also the time that these channels are available in the plasma membrane of the pacemaker cells. We know Mike McCoss has developed a nice proteome assay to test turnover of proteins, and we are trying to run some pilots with the Nathan Shock Center and see if we can incorporate that in the pacemaker tissue. Unfortunately, the pacemaker is really, really small, so the kind of things that we can do is very challenging, but I mean, the Nathan Shock, they're really good at that, so we are hopeful we're going to be able to measure that. But yeah, it's a really important factor that needs to be investigated.
All right, Darian Bug, I may have missed this, but in your last two images where you were looking at morphology, were the cells in vivo or plated?
Morphology here, probably. So this is the whole pacemaker tissue. So this is how the pacemaker looks. This is what is called the head and the tail, and these are fixed tissues. So what we do here is immunodetect a pacemaker marker that is the HCM4 channel, and we clear the tissue and we do high resolution imaging of the volumetric sample. So that's what we are looking at here.
All right, and Maria Janoska. I apologize for any name that I mispronounce. Thank you for the nice talk. Are there any other ions such as zinc that can influence how pacemaker function and dysfunction?
Wow. Yeah, there are for sure many ions that regulate ion channel and sustain the action channel potential. So potassium obviously is one that is in charge of all the repolarization phase. There is some participation of sodium channels. Zinc, that is important. I don't know the answer on top of my mind if there are specific proteins regulated by zinc that can modulate. But yeah, that's an interesting question.
All right, thank you so much. I think this is all the time we have for questions live. I would redirect everybody to the Slack channel. Actually, I'm going to put my own question over there for you. And our next speaker is, and there's an unanswered question for Anna in the Q&A tab. Anna, if you can address that. Our next speaker is Mariah Sweetwine.